Hello, welcome to Biology, the scientific study of life. Ribosomes. Ribosomes facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis. A ribosome, which is large enough to be seen with an electron microscope, is made up of two subunits, called the large and the small subunits. As is shown here in this figure, what's that? The ribosomal subunits are constructed of proteins and RNA molecules named ribosomal RNA or R small RNA in eukaryotes. The subunits are made in the nucleus. Ribosomal RNA genes on the chromosomal DNA are transcribed, and the RNA is processed and assembled with proteins imported from the cytoplasm. The resulting ribosomal subunits are then exported via nuclear pores to the cytoplasm in both prokaryotes and eukaryotes. Large and small subunits join to form a functional ribosome only when they attach to an mRNA molecule. About 2/3 of the mass of a ribosome is RNA, because most cells contain thousands of ribosomes. RNA is the most abundant type of RNA. Although the ribosomes of prokaryotes and eukaryotes are very similar in structure and function, those of eukaryotes are slightly larger and differ somewhat from prokaryotic ribosomes in their molecular composition. The differences are medically significant. Certain antibiotic drugs can inactivate prokaryotic ribosomes without inhibiting the ability of eukaryotic ribosomes to make proteins. These drugs, including tetracycline and streptomycin, are used to combat bacterial infections. The structure of a ribosome reflects its function of bringing mRNA together with the amino acid bearing its RNAs. In addition to a binding site for mRNA, each ribosome has 3 binding sites for tRNA, as it's shown here in this figure. Notice the P site. Notice here. The P site peptidyl tRNA site holds the tRNA carrying the growing polypeptide chain, while A site amino acid tRNA site holds the tRNA carrying the next amino acid to be added to the chain. Notice that. The ribosome holds the tRNA and mRNA in close proximity and positions the new amino acids for addition to the carboxyl end of the growing polypeptide. It then catalyzes the formation of the peptide bond. As the polypeptide becomes longer, it passes through an exit tunnel in the ribosome's large subunit. When the polypeptide is complete, it is released to the cytosol through the exit tunnel. 4 decades of genetic and biochemical research on ribosome structure have culminated in the detailed structure of the bacterial ribosome, which appears as a ribbon model in this figure. Notice that also recent research strongly supports the hypothesis that RNA, not protein, is primarily responsible for both structure and function of the ribosome. The proteins, which are largely on the exterior, support the conformational changes of the RNA molecules as they carry out catalysis during translation. RNA is the main constituent of the interface between the two subunits and the A and P sites, and it is the catalysts of peptide bond formation. Thus, a ribosome can be regarded as one colossal ribozyme. Building a polypeptide. We can divide translation, the synthesis of polypeptide chain, into three stages analogous to those of transcription: initiation, elongation, and termination. All three stages require protein factors that aid mRNA, tRNA, and ribosomes in the translation process. For certain aspects of chain initiation and elongation, energy is also required. It is provided by the hydrolysis of GTP, guanosine triphosphate, a molecule closely related to ATP. Ribosome association and initiation of translation. The initiation stage of translation brings together mRNA at tRNA bearing the first amino acid of the polypeptide and the two subunits of a ribosome. First, a small ribosomal subunit binds to both mRNA and a specific initiator tRNA, which carries the amino acid methionine. The small subunit then moves or scans downstream along the mRNA until it reaches the start codon AUG, which signals the start of translation. This is important because it establishes the codon reading frame. For the mRNA, the initiator tRNA already associated with the complex, the hydrogen bonds with the start codon. The union of mRNA, initiator tRNA, and a small ribosomal subunit is followed by the attachment of a large ribosomal subunit, completing a translation. Initiation complex proteins called initiation factors are required to bring all these components together. The cell also spends energy in the form of a GTP, notice that, molecule to form the initiation complex at the completion of the initiation process. The initiator tRNA sits in the P site of the ribosome, and the vacant A site is ready for the next amino acid tRNA. Note that a polypeptide is always synthesized in one direction from the initial methionine at the amino acid or the amino end, also called the N-terminus, toward the final amino acid at the carboxyl end, also called the C-terminus. Notice this as it's shown here, the initiation of translation. Elongation of polypeptide chain. In the elongation stage of translation, amino acids are added one by one to the preceding amino acid. Each addition involves the participation of several proteins called elongation factors that occurs in a three-step cycle described in this figure. Energy expenditure occur in the first and third steps. Notice that all these steps: peptide bond formation, codon recognition requires hydrolysis of two molecules of GTP, which increases the accuracy and efficiency of this step. One more GTP is hydrolyzed to provide energy for the translocation step. The mRNA is moved through the ribosome in one direction, only 5 end first. This is equivalent to the ribosome moving 5 to 3 on the mRNA. The important point is that the ribosome and the mRNA move relative to each other, unidirectionally codon by codon. The elongation cycle takes less than a tenth of a second in prokaryotes and is repeated as each amino acid is added to the chain until the polypeptide is completed. Termination of translation. The final stage of translation is termination. Elongation continues until a stop codon in the mRNA reaches the A side of the ribosome. The base triplets UAG, UAA, and UGA do not code for amino acids but instead act as signals to stop translation. A protein called a release factor binds directly to the stopped codon in the A site. The release factor causes the addition of a water molecule instead of an amino acid to the polypeptide chain. This reaction hydrolyzes the completed polypeptide from the tRNA in the P site, releasing the polypeptide through the exit tunnel of the ribosome's large subunit. The remainder of the translation assembly then comes apart. Polyribosomes, a single ribosome can make an average-sized polypeptide in less than a minute. Typically, however, a single mRNA is used make many copies of a polypeptide simultaneously because several ribosomes can translate the message from one mRNA at the same time. Once a ribosome moves past the start codon, a second ribosome can attach to the mRNA. Thus, a number of ribosomes may trail along on one mRNA. Such strings of ribosomes called polyribosomes or polysomes can be seen with an electron microscope. Polyribosomes are found in both prokaryotic and eukaryotic cells. They enable a cell to make many copies of a polypeptide very quickly. Okay, these stages, these represent elongation cycle of translation. Note that in this diagram are the proteins called elongation factors. The hydrolysis of GTP plays an important role in the elongation process. Okay, thanks for listening.